Hello students, welcome back to the official page of Read My Course. I am your biology teacher Dr. Shatabdi, and today we will be continuing in the third class of digestion and absorption, and we will be studying in details about the digestive glands. So let's begin. So these are the topics which we are going to cover today. We will have a look at the salivary glands, then the role of saliva in digestion. We will follow it up with the role of liver and what is its function in digestion. Then the gallbladder, the importance of bile in digestion. We will follow it up with the pancreas and the role of small intestine. So beginning with our first slide. First, I want you to have a very good look at the main structures which are involved in the process of digestion and what are the substances that they are producing that is finally helping in the process of digestion. So we know that saliva is produced in the oral cavity by the salivary glands that we had studied in the last class. Moving down, we have the stomach. In the stomach, the gastric juices are produced. We will read in details about the gastric juices in the coming slides. Remember that the gastric juices are produced which break down the food bolus into simpler structures, which is known as food chyme, chyme. It is a semi-liquid or semi-fluid simpler form of food which can be easily absorbed farther down in the intestines. Right, then we have the bile is another important substance which is produced by the liver. Remember, it is produced by the liver and it is stored temporarily in the gallbladder. Okay, so if somebody asks you where is the bile produced, it is produced in the liver. Remember, not in the gallbladder, it is stored in the gallbladder. Okay, after that we have the pancreatic juice. The pancreas is a structure which produces the pancreatic juices which further again help in digestion. How they help, we will read in the following slides, in the coming slides. And we have the intestinal juices as well. So this is a whole, these juices come together, these enzymes come together and they stepwise break down the food into simpler forms until and unless they are broken down into such simple units, small units that can be easily absorbed into the bloodstream, right? With that, we move to the next slide. So we have already read in our last class that there are three major salivary glands. What are they? The parotids, which are the largest. Then we have the submandibular. Then we have the sublingual. Right? The location of parotid is in the upper part of the cheek, just in front of the ears. Okay. The location of the submandibular, as the name suggests, mandible is the lower jaw. So the submandibular glands will be located somewhere below the lower jaws. Right? And we have the sublingual, that is just below the tongue in the floor of the mouth. And they are paired, one on right, one on left. That much is clear. Okay. Now see, parotids are the largest and they specifically produce an enzyme which is known as the ptyalin or the salivary amylase. Remember the name ptyalin salivary amylase? This particular substance is responsible for initiating the process of chemical digestion, that is breaking down food into smaller or simpler particles. Right? What it does is it breaks down the complex form of starches into simpler forms like glucose, like maltose, right? That is, polysaccharides are broken down into disaccharides and monosaccharides, and this process is initiating where? In the oral cavity. How? By the action of the salivary amylase or ptyalin from the saliva. Right? Moving to the next slide. So we have already studied this about the alpha amylase or the ptyalin. Then we already know saliva initiates the process of chemical digestion. Remember one fact that approximately 1.7 liters of saliva is produced on a daily basis in the oral cavity, right? Another important secretion by the saliva is mucus secretion. What it does is, though it does not directly help in the process of digestion, but what it does is it coats the food or lubricates the food substances so that it becomes easy for us to swallow the food, right? So indirectly it helps in swallowing of the food, deglutition of the food, right? So moving to the next slide, we come to the stomach. I want you to have a look at the functions of the stomach first. See, we know that it stores the food temporarily. It is a sac, J-shaped sac, which stores the food, right? What is the role of stomach in digestion? One is mechanical, that is churning of food. What happens is the stomach walls have muscles. We know that the stomach walls have two types of muscles, the circular muscles and the longitudinal muscles. So these muscles, wave like contractions occur. Okay, these wave like regular contractions and relaxations, what it does is it mechanically moves the food inside the stomach and breaks down the larger food bolus into smaller particles, right? After that it becomes ready for chemical digestion. So first of all, what it does is it churns the food, right, and breaks them down mechanically into smaller particles. After that, what happens is chemical digestion. And the most important part of stomach in digestion is that it helps in protein digestion. Specifically, it helps in protein digestion. It has proteinase enzymes which help in protein digestion. So remember this fact that the gastric glands that line the stomach wall, the innermost wall of the stomach, was the mucus layer, mucosa, right? So they have small openings which contain the gastric glands. They produce a acidic solution. This acidic solution helps to lower the pH of the stomach. Why it is important to lower the pH of the stomach is because we have to activate an enzyme which is produced by the stomach, that is pepsinogen. This pepsinogen is the inactive form of the enzyme which gets activated into pepsin. Now, until and unless the environment inside the stomach is acidic, this pepsinogen, which is the inactive form, will not get changed into pepsin, and it will not be able to act on the food molecules. So that is why it is important that the environment inside the stomach is acidic. Now, a question may arise in your mind that if the environment is acidic, then how come the lining of the stomach is protected from these strong acids, very strong acids like hydrochloric acid? The thing is that they have the mucus lining on the walls of the stomach. This mucus lining separates the wall of the stomach from the acid inside the stomach, right? I think you get it. And that is why the lining of the stomach is protected against the corrosive action of the strong acids, that is the HCl. So what is the normal pH inside the stomach? It's highly acidic, 1.5 to 2. Okay. With that, we move to the next slide. I have already said that stomach is responsible for chyme formation. What is this chyme? I have described. It is a semi-fluid form of food, which is a simplified form of food from the bolus which we had swallowed through the mouth, right? Now, I have said that the stomach produces gastric juices. The gastric glands which are lining the wall of the stomach, they produce this gastric juice. What is the main component of the gastric juice? We have HCl, we have pepsin, right? HCl, why it is important? It lowers the pH, it makes the pH 1 to 2. What happens is it kills the bacteria and denatures the proteins, right? Pepsin is basically an enzyme and it helps in further breaking down of the proteins. So stomach is one of the main sites of protein digestion. Remember this, okay? So there are different types of gastric glands which are present in the lining of the stomach. What are they? One important is the chief cells. The chief cells, what they produce is they produce pepsinogen and they produce the gastric lipase. Pepsinogen, we know that pepsinogen gets converted to pepsin and finally help in the breakdown of proteins. What about gastric lipase? Gastric lipase basically helps in the breaking down of fats or lipids. Okay. Then we have the parietal cells which basically produce the hydrochloric acid. And we have the mucus cells which secrete the mucus, which separate the corrosive acid from the lining of the stomach. Okay. With that much information, we are moving to the next slide, that is the liver. So first we have to know some facts about the liver. The liver is the largest gland in the body, right? Remember this. Where is it located? In the abdominal cavity, just below the diaphragm. Okay, and it is lobulated. The liver has various lobes. And the cells of the liver are known as hepatocytes. Hepatic cells, they are known as hepatocytes. And these hepatocytes are the structural units the functional units of the liver. And what happens, what they do is they produce this bile. Okay, this bile is, I have already said, it is a yellowish green fluid which is produced in the liver mainly by the hepatocytes of the liver, and their major function is nothing but digestion of lipids and fats in the small intestine. Digestion of lipids or fats in the small intestine, that is the major function of bile. Okay, moving to the next slide, we have the gallbladder. See, I already told you bile is produced in the liver, stored in the gallbladder, right? Another important function of the gallbladder is it concentrates the bile which is produced by the liver and makes it ready for the digestion, right? Bile also has another important function, that is it helps in fat emulsification, that is breaking down the larger fat droplets into smaller droplets which can be acted upon by enzymes further. Okay, so fat emulsification is an important function of bile. So with that, moving to the next slide, I want you to have a good look at this picture, because here what I am going to show you is the ductal system or the duct systems which are involved. See, okay, this is the gallbladder, this green structure, right? We have a duct from the gallbladder. We have two ducts from the liver. This duct from the gallbladder and from the liver, they join, and what they form is this common bile duct, right? This common bile duct is opening into the first part of the small intestine, duodenum, we have already read. Okay, pancreas. This pancreas has a pancreatic duct, right? Now, when this pancreatic duct is joining with the hepatic duct, what we are getting is this hepatopancreatic duct. So it is a branching, like the branches of a tree. So it is a branched system of ducts. Starting from the gallbladder, we have the hepatic duct, and we have the duct from the gallbladder joining to form the common bile duct. Then the common bile duct joins with the pancreatic duct, and they form the hepatopancreatic duct, which is opening ultimately into the duodenum, which is the first part of the small intestine, right? With that, we move to pancreas. Pancreas acts as an exocrine organ and an endocrine organ. Okay, exocrine organ means the substance which it is producing is being produced somewhere else, then it is traveling via ducts and it is getting released into somewhere else for its function. Okay, that is the meaning of exocrine. And endocrine means the structure that is producing the enzymes or hormones, that the function occurs locally. There is no ductal system involved. That is the main difference between exocrine and endocrine. Now, how the pancreas acts as an exocrine and endocrine system both? See, basically the exocrine portion of the pancreas, it produces the pancreatic juice, right, which gets transferred into the small intestine via the pancreatic duct, which we had read in the last slide. Also, the endocrine function is that how it acts as an endocrine gland. It secretes hormones like insulin, glucagon, etc. They do not travel via any ductal system. They act locally. I hope you get it. That is why it is both an exocrine and an endocrine organ. It has both functions. Okay. So, what is the main secretion of the pancreas? It is secreting, just have a look. It is secreting bicarbonate ions. Okay. And what is the main function of these bicarbonate ions? They are neutralizing the acidic pH of the stomach. The food that is being released from the stomach is acidic in nature, mainly because it was in an acidic environment. So what the pancreas does is it releases bicarbonate ions and makes the environment alkaline, right? Also, it secretes lipase for fat digestion, amylase for carbohydrate digestion, and proteinase, for example, trypsin for protein digestion. So, trypsin is produced in the small intestine, remember. There is no trypsin production in the stomach. Okay. Moving to the next slide. What about the small intestine? Succus entericus. Succus entericus is the intestinal juice. The name of the intestinal juice is Succus entericus. Okay. It is a intestinal juice. It is a fluid which is secreted by the cells which are produced in the small intestine. It is again alkaline in nature, and it helps to counteract the high acidic and proteolytic chyme entering from the stomach further. Right? So, this is only about the digestive glands and what their secretions are. Now, how they break down the food stepwise, that we will read in the digestion of food. That will be coming up in the next sessions, right? So, that brings us to the end of this class. In the next class, we will be reading about digestion of food, the stepwise how food is broken down from the complex substances into simpler substances. And remember one fact, that the human stomach has a volume of 1 liter approximately in adults, okay? But when we have a hearty meal, they have the capacity to distend themselves or expand themselves to occupy almost 4 liters of volume of food or whatever substance we are taking in. So this is mainly possible because of the muscular layer of the stomach, which has the ability to undergo distensions or expansions. But of course, they have a limit, right? So that is an interesting fact you should know about the stomach. And with that, we come to the end of this session. We will meet in the next session where we will be studying in details about the digestion process. Okay, till then, stay safe, guys. Take care, stay alert, stay at home. If you like this video, then please do like, share, subscribe, comment in the comment section below. Let us know how we are taking your classes, whether you are liking them or not. What are the improvements which can be done? If you want to ask me any question, please do feel free to ask. I will try my best to communicate with you. And till then, bye guys.